So, to recap from Lecture 1, many dinosaur bones are not fossilized. Many? You mean none? Ooh, in 2005, Mary Schweitzer surprised scientists when she reported finding soft tissue in dinosaur bones. She started a firestorm of controversy in 2007 and 2008 when she reported sequencing proteins in dinosaur bones. Critics charged that the findings were mistaken and that the soft tissues was just biofilm created or produced by bacteria that enters outside of the bones. Schweitzer answered back with testing with antibodies. Her report in 2009, the presence of collagen and other proteins that bacteria don't make. The biggest reason for the controversy is because the presence of any tissue should have been gone and can't even have lasted more than 1 million years under the right or the best of conditions. Likewise, if these dinosaur bones are 65 million years old, there shouldn't be any tissue or carbon-14 within them. That's saying, when you test something for carbon, like in a dinosaur bone, the results should come back 0. None. Carbon-14 testing will or will not show.
Big picture. Evolution holds that dinosaurs evolved 220 million years ago and died about 65 million years ago from any of a number of possible extinction events. But the Bible teaches that all land animals, including dinosaurs, were created on the sixth day of creation, just thousands of years ago, and they were all named by Adam just before God handed over his completed creation to Adam and Eve, charging them to take dominion over his completed creation. Then, about 4,400 years ago, the entire world was deluged by Noah's flood, and the dinosaurs, along with billions of other creatures, were wiped out. Let's take a quick look at some evidences that show that the biblical account might actually be true. First, dinosaurs are cleverly designed. Consider this Triceratops. Its 2,000-pound head is mounted in a way that allows it to turn every which way, yet still be strong enough to ram something, even while running. What about massive sauropods with weight-bearing systems from hip to toe that allow its 200,000-pound body to even walk, and neck vertebrae that are 90% filled with air so it can lift up its head? Next, there's the absence of dinosaur ancestors and transitions. Even the Chicago Field Museum sign admits there have been 0 transitions between dinosaur kinds. It shows question marks regarding where they came from. It's almost like someone just put each basic kind on Earth right at the same time. There are even dinosaur design features that show they lived in the ideal pre-flood world. Pterodactyls may have been too heavy to fly in today's atmosphere. Same with huge pre-flood dragonflies. The fossil record is filled with giant creatures and plants. We know this because there are billions of dead things buried in rock layers laid down by water all over the earth. And when we look at the dinosaur fossil record, we see that they were buried furiously, rapidly, and simultaneously, oftentimes found fleeing in groups. Take this massive bone bed in Hilda, Canada. Thousands of centrosaurs were catastrophically buried over an entire square mile. Or this one in China, where thousands of different kinds of dinosaurs were simultaneously buried in a single 980-foot-long ravine. There are hundreds of dinosaur bone beds all over the world, including the U.S., where the Morrison Formation covers 13 states and 700,000 square miles. Thousands of torn-apart dinosaurs are buried here in hundreds of mass graves, with many found in the classic death pose with their necks arched back, choking as they died. Museum signs everywhere even admit they died in a watery catastrophe. Some dinosaurs are even found mummified, with tree leaves, flowers, ferns, shrubs, and algae still in their stomach. Dinosaurs are even found buried with marine creatures. Isn't a global flood the best explanation for this? Scientists have been discovering soft tissue in dinosaur bones. They describe blood cells, blood vessels, connective tissue, and even collagen, which has a maximum shelf life of about 900,000 years at 40 degrees. With a maximum shelf life of less than 1 million years, what's collagen doing in dinosaur bones that are supposedly 65 million years old? Many dinosaur bones are even found unfossilized in places like Madagascar, Alaska, and Montana. Does this evidence seem to support the Bible's recent and violent flood, or evolution's story?
Okay, sorry. It has been pointed out many times that fragile, complex molecules like proteins, even in hermetically sealed, should have fallen all apart by themselves from thermodynamic considerations. along and well under 65 million years old, that evolutionists insist have passed since. Many anti-creationists, therefore, breathe a sigh of relief when in mid-2008 a paper claimed to have found evidence that transparent blood vessels, for instance, were the result of recent bacterial formation of biofilm forming that followed the shape of formation of, wait, hold on, woo, got a little mess up there, got you there, buddy, that followed the shape of, oh, there we go, here she is, that followed the shape of, well, where the origin vessels lie, and that the red blood cells are actually iron-rich spheres called framboids. There were substantial reasons why not just creationists, but Schweitzer and other non-creationists were not all convinced by these claims. Well, let's look at the new findings. Yeah. Nate the Great here. Now comes a further announcement by Schweitzer and others in the prestige journal Science. A substantial additional evidence to bolster her previous findings. A specimen on this occasion was a piece of fossil Hadrosaurus, duck-billed dinosaur, regarding by evolutionary assumptions as being 80 million years old. In short, the researchers found evidence of the same fibrous matrix, transparent, flexible, very flexible, vessels, and preserved microstructures she had seen in the T-Rex specimen. Only this time, they went to exceptional lengths to isolate critics. Got it. Isolate those haters. Haterade. Critics say that her claims were given the millions of years perspective are indeed extraordinary. Require extraordinary evidence. And that has been amply provided. Yet the critics demand additional protein sequencing, super careful handling to avoid claims of contamination, and confirmation from other laboratories. So Schweitzer and her team set about doing just that when they looked at the leg bone of the Hadrosaurus encaged in sandstone. Extraordinary measures were taken to keep the sample away from contamination until it reached the lab. They used an even more sophisticated and newer mass spectrometer and sent the samples to 2 other laboratories for confirmation. They reported finding not just collagen but evidence of 2 additional proteins, elastin and lamin. They also found structures uncannily resembling the cells found in both blood and bone, as well as cellular base membrane matrix. And there were, once again, hints of hemoglobin. From applying hemoglobin-specific antibodies to the structures and seeing if the antibodies would bind to them. Even leaving outside the hemoglobin, Schweitzer's paper is huge news. Indeed. Okay. So, look, I kind of do agree with this. I mean, maybe I'm the only one in the classroom. I don't care if I am or not. But the philosophers of science has written much about the power of paradigms, especially when it has worldwide implications, such as long age belief. Such a paradigm is seldom, if ever, overthrown simply because of observation that contradicts its expectations. Well, they would say different. Even Schweitzer herself, despite professing to be an evangelical Christian, which would make sense, although she was mocked before because she had a biblical scripture in her office and she said she was a Christian, but believed in a type of evolution. I believe in evolution too. I believe in micro, not macro. All right, let's just get that straight, damn it. So, anyways, it is extremely defensive about the old age paradigm. What happens is that hypotheses and assumptions are constructed to preserve the intactness of the core hypothesis. In this case, what is known as a deep time. In simple terms, proteins should simply not have been able to last these tens of millions of years. So when they are found in specimens dated this old, the paradigm is under serious threat. Yes, I would have to say it would be under serious threat too. I mean, considering that we have these millions of year old dinosaur bones, as we say, fact scientifically proven that they are, but yet we trip along some tissues and other things that might say, maybe we should re-question these dinosaur bones.
Just in the last 20 years, over 50 articles have been published in scientific journals that have documented 14 bioorganic materials in dinosaur bones that simply cannot be millions of years old. These include blood vessels, red blood cells, hemoglobin, bone cells, ovalbumin, chitin, unmineralized bone, collagen, limited DNA, skin pigments, FEX proteins, histones, keratin, and elastin. Just 20 years ago, most scientists simply assumed dinosaur bones were solid mineralized impressions of bones. But modern science has now revealed that many of the dinosaur bones are original, even at the molecular level.
Hello, I'm Neil deGrasse Tyson, your host for Nova Science Now.
For the last decade, the African island has offered Ray and Christy Rogers a spectacular excavation site. They work in an area of remote grasslands where dinosaur bones literally cover the ground. An ancient graveyard where an unusually large number of dinosaurs died and were entombed by mud flows that preserve their bones.
The preservation is unbelievable. The quality of the material is exceptional. This is a piece of bone from the Madagascar locality. It, in many regards, doesn't even look like a fossil. This is typical of the Madagascar material. It is not heavily stained. It's not been invested with iron, with manganese. It doesn't look like a typical fossil. If you didn't know, you might think you're picking up a cow bone today, a bleached cow bone.
I was looking at this thing thinking, this is really interesting.
It was the inside of the T-Rex bone that fascinated Schweitzer, who made thin sections out of it. And in these cross-sections of fossilized bone, she saw something that she and everyone else had thought was impossible. Round structures that looked like red blood cells. Dinosaur blood cells.
Inside those channels where the blood vessels would have run were these little round red structures that were all kind of lined up like a train, and they were bright red and translucent. Nobody else had seen anything like that before.
The very idea of blood cells in a 70 million year old bone was more than unconventional. It was radical.
Nobody was imagining that dinosaurs might have had preserved soft tissues.
Derek Briggs is curator of invertebrate paleontology at the Peabody Museum at Yale University.
So along comes Mary Schweitzer, and she's starting to look inside dinosaur bones and has made this startling discovery about the presence of red blood cells. What was your initial reaction to that?
Oh, I think the same reaction as everybody's, that this was totally improbable. She perhaps misinterpreted the evidence or was exaggerating the potential for what she was seeing.
So skeptical at first.
Oh yeah, definitely.
Why do you think it didn't occur to anybody?
Because we have this clear understanding that part of all biological cycles involves decay. I mean, nature is set up to break down that material and recycle it. So it's just improbable that those kinds of very delicate structures would survive, particularly for millions of years.
When you think about it, the laws of chemistry and biology and everything else that we know say that it should be gone. It should be degraded completely. This is not possible. Do it again. We got another piece of bone. We put it in the solution. We waited 2 or 3 or 4 weeks. Looked again. More blood vessels. We must have repeated that with probably 17 or 18 different fragments of bone.
Well, it's time for me to look nuts. See how nuts I can look. I like looking nuts. It is commonly inserted that the fossil record provides the best evidence for evolution happening on Earth. It is important to remember that the geological evidence is the same for everybody. We all observe it. We all see it. It is an interpretation that differs. What do I mean? Well, we can say that these layers took millions of years old. Or we could just say something catastrophic had happened that made the layers happen and form the way that they did. We have to conclude with asking, why are there preservations? Why is these bones well preserved? They really shouldn't. The fossil record is no different. The evidence exists in the present. Yes, it exists in the present. And the interpretations are about the past. No fossil, rock layer, or dating method can ever prove that evolution did or did not happen. The same is true for creation. Let's not recall. What's just nuts, alright? The presupposition used to interpret the evidence will affect the conclusions. The fossil record must be interpreted. It cannot speak for itself. A belief in the story of geologic time is a fundamental idea in all evolutionary contexts. If the vast expansion of time suggests by uniformitarian geology did not occur, evolution cannot occur. Most people, including those with training in the science, are not aware of the many assumptions that radiometric dating has with the geological column and the fossil interpretation used for the foundation. Biologists assume that geologists have correctly identified the age of rocks. The geologists assume that the chemists have correctly identified the half-life of the different isotopes. Chemists assume that the physicists have correctly identified the details of radiometric. Wait a minute, you're dumb, bro. It's radioactive decay. This chain of assumptions supporting evolution brings down the entire structure if any one of these chains is broken. So you got point A to point B. Nothing can be disruptive between point A and point B. It has to be point A and point B. It has to be 2 plus 2 equals 4, not 2 plus 2 equals 2. But if any of these are, from somehow you get to point A to point D, maybe B, C, and D, if any one of these letters, even the simple A, B, somehow I can't get to B, somehow I can't correctly say all B, something's wrong. So if there is something wrong, like there is a problem with me saying B, because you have B being a bone that has tissue in it. well, then you have a problem. So if you have a problem getting to A to B, is probably because within all these chains of events, of scientific events, showing, look, we can conclude all these, it is factual. You cannot have one that's interrupted with these bones, because if these bones really have tissue, we're in problems. We're in deep problems. No, wait. Because according to Schweitzer, she says, we have explanation. This makes sense. Tissue can be in these bones to perfectly match the time scale of what we give these dinosaur bones. Yes, she says she solved it. So technically, we knew they were in the bones. They were in the bones the whole time. It's thinking. We knew that. We're just throwing a monkey wrench in and just get you guys startled up and everything. Like you didn't even know what you're doing. Yeah, nice one. So, here's the thing. So, her conclusion is, hey, we understand all this. It makes sense. They would be in the bones. Hello. There's like iron in it. The iron preserved the whole structure of what we're seeing. Iron. Well, yeah. I mean, you know what? Let's just have an expert explain this. Who am I? I'm just a messenger. I'm just a reader. I don't really know all this. I'm not a physicist. I'm not a biologist. But hey, I guess it takes a dummy to critically think a dummy, huh? So anyways, I'll have somebody that's more of an expert that actually ran a laboratory, $1 million laboratory. This guy knows what he's talking about. I'll actually have him go into great detail about this tissue and the bones, the preservation, if iron actually can preserve something 80 million years old or even 65 million years old, and we'll just have him break the science down. Listen, this guy. Wait, don't go down. Just wait. I forgot to add something. So, in a conclusion, scientifically, like any scientist finds anything that has to be published, they actually have to explain their findings. And so the way that Schweitzer actually went in and to deduce and actually conclude her actual findings is that she actually used an ostrich and a chicken. Yes, their tissues. Yeah, because they say that it's closer to a dinosaur, because they believe in an evolution that they say they're finding more evidence that actually wraps that dinosaurs actually came from birds. That birds are closer to dinosaurs. Yeah, that's cool, right? T-Rex, little guy, little stubby arms. Yeah, he had wings. Well, maybe how? Well, maybe his descendant. Maybe, you know what? He did it. But dinosaurs say, it don't even make sense. You know what? It don't even matter. So what? Dinosaurs used to fly. T-Rex used to fly. His little cousin over there used to fly with his little arms and stuff. Well, anyways, it don't even matter. That don't even matter. So, anyways, just wanted to add that an ostrich tissue and chicken tissue was actually used to try to explain the preservation of this tissue. And that's found in dinosaur bones. So, hope you like it. Bye. Boop.
If you cheated by looking at my answer. And so to help you, here's a little diagrammatic representation of a protein going through aldehyde fixation. Tell me what all these little bars are, all these little crosslinks. I want to know what they are. You're talking about iron nanoparticles. Now we're talking things that are nanometer sized. In fact, some of these have measured out about 100 nanometers in size, these iron nanoparticles. And according to Schweitzer, they perform the fixation. They cross-link the proteins. Now, again, I mentioned that the molecules that we use in fixatives to fix these proteins are angstrom-sized. And the reason they are able to fix these proteins is because they can surround the entire protein because they're angstrom-sized, and they can fix the tissue by that cross-linking process. And so you're expecting a nanometer sized, in fact 100 nanometer sized iron particle to do the same fixation that an angstrom sized molecule does. It's kind of like using a trash truck to staple your papers together. Okay, you're using a trash truck to staple and immobilize your papers that are fluttering around in the wind. And this trash truck is going to staple them together so that when you pick them up, they're held by a staple and they don't move around. They're cross-linked. Okay? And so nanometer size, 100 nanometer size iron particles don't do the trick. And again, I'm waiting to hear your answer, looking on my email to see if you can answer that. So you have an abject ignorance of the fixation process. And so I would advise you to look into the protocol manuals and study them. Now let me make another point about this supposedly fabulous system of iron preservation of tissues. If iron preserved the tissues for 70 million years with the kind of stunning preservation that I'm photographing from these dinosaur soft cells, if iron could perform that, then why isn't it in my protocol manual? Okay? I'm a member of the Microscopy Society of America, have been for many years. I'm on the board of directors for the Southern California Society for Microscopy. Nobody that comes to present at our meetings has ever talked about iron preservation of tissues. And so if it is, as you say, a fabulously efficient fixation protocol that can fix proteins and tissues for 70 million years, then they should be in our protocol manuals. In fact, I should be able to find it in the latest edition of Weider's Functional Histology, which is the textbook for studying histology and tissue relationships. And of course, this goes into great detail to talk about fixation. So why isn't iron preservation in this textbook? Why isn't it in any of the textbooks or protocol manuals that we use for microscopy? Number one. Number two, why aren't the cryopreservation companies using it? You could run down and find a patent attorney, my friend, my critic friend. Run and find a patent attorney and get yourself a patent pronto for iron preservation of tissues, because the cryopreservation banks would buy that from you and you'd be a multi-billionaire. Okay? So go get your patent if this is such a hot thing and it really works the way you say it does, because it doesn't. Now let me just make one more point before I do a review here. And that point is this. When you look at Schweitzer's explanation, her theory that iron preserved the tissues, and you study the materials and methods section, which is a little difficult to find, but if you go and find it and you study it, you realize that Dr. Schweitzer had to first use an anticoagulant to prevent the blood from clotting. Why did she do that? She put an anticoagulant in the blood that was used, the chicken blood and the ostrich blood that was used to soak the tissues in, because if that blood coagulated, it would have bound up the iron and made it unavailable for her experiment. She needs the iron. She has to have the iron free and available for the experiment. And so she first has to put an anticoagulant in there to prevent it from clotting, number one. Number two, when you read the materials and methods, you find that she used ultracentrifuges multiple times during the experiment. Why? Well, she had to filter out all the serum. All the serum that was full of clotting factors and other factors that bind up these proteins tightly in the clotting reaction. She had to remove the serum with all those clotting factors. She had to remove all the cells, all the platelets and subplatelet cells and other cells, white cells related to clotting of blood and of tissues to form scabs. All of that had to be removed through multiple series of ultracentrifuge use. And finally she had to use molecular filters to filter out these things. And so she spun everything down multiple times, and finally she was left with the red blood cells, which she broke open. It's called lysed. She lysed them open, and that, of course, freed the hemoglobin, which has the iron in it. Now, none of those conditions that can be said are indicative or related to what happened at the Hell Creek. Okay? When those things were buried at Hell Creek, There were no ultracentrifuges. There was no anticoagulant available. There were no molecular filters. And so just to get to the point where she could begin the experiment, she had to employ all that laboratory technology. And then, of course, she left it on a laboratory bench at room temperature for 2 years in the absence of water, which is abundant at Hell Creek, the freeze-thaw cycle, the heating of the Montana summer, and all of the microbes and scavengers and insects and everything that was fully involved in those specimens, and particularly my specimen, the Triceratops horn, fully open to the environment, highly vascular, and so she had to remove all that from her experiment in order for her experiment to work. And so all one has to do is look at the current issue of Microscopy and Microanalysis. This is the December 2015 issue, and a wonderful little paper in here which studies collagen deposition during skin repair in rats, and it goes through all of the clotting factors, all of the proteins and factors that are involved with this fabulous mechanism of clotting that had to be removed in order for that experiment to proceed. And so in review, my critic has shown that he can set up a straw man. Armitage has no independent supporters, and then knock it down as if that's some big claim against me and my work. He also talks about how Schweitzer says that young Earth creationists are hijacking her scientific finds. My friend, I did my own work. Okay, I went to the Hell Creek. I found Triceratops horn, Triceratops frill, Triceratops condyle. I found Nanotyrannus vertebra and Nanotyrannus metatarsal, and there are cells in all of them. I'm not hijacking anybody's work. I'm doing my own work, and I'm publishing it successfully. And so, for you to set up a straw man and knock it down shows just how desperate you are. Next, you showed an abject ignorance of the effect of free radicals on phospholipid membranes in cells. You show an abject ignorance of the fixation process and how angstrom-sized molecules do the fixation and not these 100 nanometer sized iron filaments. By the way, here's the answer to the question that I've been waiting for you to answer. In fixation, it is the amine groups that are cross-linked in proteins. Now, that leaves the whole rest of the protein, the non-amine portion of the protein, available for degradation and decay. And so, once again, if iron preservation can preserve these structures, beautiful, fabulous structures, for 70 million years, you need to go out and get a patent and get yourself rich, buddy, because you got something that nobody else in the world has. And so